Glycolysis is great. We can break down glucose to give us pyruvate, and in the process, we get ATP. We get 2 ATP directly from glycolysis, and then we also get this NADH, these 2 NADH, which is where we often are going to get the bulk of our ATP generation is actually by using that NADH, taking it through the electron transport chain, using oxidative phosphorylation of the electron transport chain in order to generate ATP. So remember that from each NADH, we are able to generate like 2.5 ATP. So most of the ATP that we could generate from glycolysis is actually going to come from that NADH, but we do generate ATP directly from glycolysis. And that's better than nothing. So if you can't do the electron transport chain, then you're going to want to keep glycolysis going. But in order to keep glycolysis going, whether or not you have the electron transport chain, you're going to need this NAD+. So remember that in glycolysis, in this glycerol-3 phosphate dehydrogenase step, because we're doing this oxidative phosphorylation, we're adding this whole phosphate group, including the oxygen, not just adding a phosphoryl group like we do with this kinase step. We're doing this oxidation, we're going to need NAD+. And if we don't have NAD+, we can't do this step. To make things even worse, if you have a lot of NADH, so remember NAD+ and NADH are just two sides of the same coin. So if what you're saying is you don't have enough NAD+, you're saying you have more NADH, and that NADH is actually going to directly inhibit metabolic enzymes involved in catabolism. So remember, like the breakdown of molecules and glucose breakdown is glycolysis, and glycolysis is inhibited by NADH. More specifically, this kind of last step, this pyruvate kinase is going to be inhibited by NADH, as well as the step that takes pyruvate and oxidatively decarboxylates it to give you acetyl-CoA, which can then enter the citric acid cycle. So NADH is going to inhibit that. It's going to inhibit a lot of other catabolic pathways. Bottom line, we don't want NADH building up. And so we need to have a way to regenerate that NAD+, not only so we can keep glycolysis going, but also so that we don't inhibit those other processes. This is assuming, however, that our goal is to produce more energy. And so you can think, well, if you have NADH, you have a lot of energy, then maybe you don't want to be doing this pathway anyway. And so then it would make sense to inhibit it. But if we do want energy, we do want to keep glycolysis going, we've got to regenerate that NAD+. If we have oxygen, our best bet is to go through the electron transport chain in the mitochondria, pass those electrons from NADH off to complex 1, which passes on to Q, and then that'll regenerate our NAD+. That'll then also, those electrons, the passing of those electrons will be used to generate ATP. And remember, we can get about 2.5 ATP per NADH produced. And so that's great. That's a great way in which we can regenerate NADH. We'll talk, however, about there's a complication where you can't actually take NADH from the cytoplasm. So where glycolysis occurs, you can't directly take that NADH in. So we'll talk about a couple of shuttles, the malate-aspartate shuttle, which is going to involve our friend malate dehydrogenase. It's going to involve both the cytoplasmic and the mitochondrial versions of it. And we'll also talk about the glycerol-3 phosphate shuttle, which is going to use glycerol-3 phosphate dehydrogenase, mitochondrial, and cytoplasmic versions of it. And this is going to allow you to kind of sneak the electrons from NADH into the mitochondria to go through the electron transport chain, but the NADH itself is not going through. So you're going to be regenerating the NAD+ out in the cytoplasm where you need it for the glycolysis. One way or another, you're going to be regenerating that NAD+, but that process is only going to take place if you have oxygen, because that electron transport chain is going to require the oxygen. And so if you don't have the oxygen, you're not going to do the electron transport chain. So you're not going to take those electrons into the mitochondria to go through the electron transport chain. If we can't do aerobic respiration, there are options to anaerobically ferment. So anaerobic in the absence of oxygen, we can do this process called fermentation. When we talk about fermentation, we're regenerating NAD+, so we can keep this cycle going without using electron transport chain and oxidative phosphorylation and all that good stuff. In our bodies, we do lactate fermentation, and we'll get a lot more into this process. There are also organisms that do alcohol fermentation and that are actually able to make ethanol from that pyruvate they generate from glycolysis. And in that process, they're going to regenerate the NAD plus in order to keep glycolysis going. What we'll see in common with all of these things is that we're having some sort of reduction reaction after we end glycolysis. So remember, glycolysis is an oxidative reaction. And so if we want to regenerate that NAD plus, we're going to need to oxidize the NAD, which means we need to reduce our substrate. So we do some sort of reduction reaction to that pyruvate in order to regenerate our NAD plus. So this is showing it in the stoichiometry, like per 1 glucose, you get 2 pyruvate. In some bacteria and yeast and plants, they go through this alcohol fermentation. They take that pyruvate, they decarboxylate it, so they remove this CO2 group. This gives you acetaldehyde, and that CO2, well, that's what's going to make your bread rise and things, because that's going to escape as a gas. This acetaldehyde, so that step wasn't a reduction, but now you can reduce it with alcohol dehydrogenase, and this is going to give you your ethanol. We don't have this pathway, but we do have another option for fermentation. The option that we use is to make lactate. So just a quick note is that lactate and lactic acid, those are just the conjugate acid and conjugate base forms of the same molecule. So see both terms used, and they're just readily interconverted by giving or taking a proton. Remember that this giving and taking a proton is not going to be redox. Remember, a proton is just a proton, not an electron, and so it's leaving behind the electron, so it's not going to change the oxidation state of anything if you have a deprotonation. So lactate to lactic acid is not redox. However, pyruvate to lactate is. More specifically, you're reducing pyruvate, oxidizing NADPH in the process to regenerate your NAD plus that then you can use to power glycolysis. This is often used by muscles that are working really hard, so they don't have the oxygen that they need to take that NADH and take the electrons from it through this electron transport chain to generate ATP. And it's also going to be used by cells like your red blood cells, which you don't have those mitochondria, and so can't do the electron transport chain. You wouldn't want that lactate to build up. Now, lactate, it's a myth that lactate actually causes your delayed onset muscle soreness, but it can inhibit your muscles in the moment if you're building up a lot of it. And also, you are going to be wasting a lot of fuel if you just have a bunch of lactate build up. Because remember that most of the energy we get from the breakdown of glucose is actually going to be happening because of the NADH and the VDH2 that we get after the steps of glycolysis. So we don't want to waste all that energy that's still stored in the pyruvate, or at this case, stored in the lactate. And so we're going to ship it off to that lactate off to the liver. When this happens from lactate from your muscles, we call it the Cori cycle, but something similar is happening with your blood cells and other cells that are doing lactic fermentation. What's going to happen is basically in your muscles, you're generating that lactate. And this is through this enzyme lactate dehydrogenase. So this LDH is converting your pyruvate to your lactate. This lactate can travel through your bloodstream. It's going to travel to your liver. In your liver, now you have another lactate dehydrogenase. So you had a copy of it in your muscles and a copy of it in your liver. Well, you have lots of copies of it. But basically here, you're going to run the reaction in reverse. So in your muscles, you were reducing pyruvate to make lactate and using NADH and regenerating NAD plus. In your muscles, you're going to be taking lactate and you're going to be oxidizing it to form pyruvate. So this is taking NAD plus and generating NADH. This pyruvate then, if you wanted, you can go and use it to make ATP, or if you want to basically use this as a cycle. So remember, when we're talking about a cycle, we want to keep things going without having to add in more stuff. In this case, what we could do is we could take that pyruvate and we could actually use gluconeogenesis to make glucose. So remember that your livers have the machinery for doing gluconeogenesis, so for actually making glucose from pyruvate. And your other cells, except for like some parts of your kidney and stuff, they don't have the ability to do gluconeogenesis. So your muscles couldn't make fresh glucose from that lactate or from that pyruvate, but your liver cells can. When we do gluconeogenesis, we're now able to make glucose. And yes, we're going to have to put energy in to make that glucose, but we can do it in the liver, and it's still worth it because we get this glucose that we can then ship back to the muscles, giving the muscles a fresh source of energy so they can keep going. And it's great that we're getting NADH because we're going to need that for gluconeogenesis in order to reverse the step that we needed NAD plus for in glycolysis. So that glycerol-3 phosphate dehydrogenase step, in the gluconeogenesis direction, we're going to need NADH. And in the case that we have here, we're actually generating that NADH in the cytoplasm. So it's ready to use, but this gets us back to thinking about what if it's not ready to use? And this gets us into thinking about how malate and oxaloacetate can be used to kind of relay electrons. And this will help us understand our NADH electron shuttles. So let's review this, and then we'll go and we'll look at those shuttles. Going back to when we were talking about gluconeogenesis, remember how we kind of talked about how there was this first part where you go from pyruvate to oxaloacetate to phosphoenolpyruvate, how you have to go through those two steps, so the pyruvate carboxylase step and the pep carboxykinase step. And you had to do the pyruvate carboxylase step in your mitochondria, but you could do the pep carboxykinase either in your mitochondria or in your cytoplasm. If we get that pyruvate from lactate, we're going to be generating that NADH in our cytoplasm. And the cytoplasm is where we're going to need it to reverse that step that we needed the NAD plus for glycolysis. However, if we're getting pyruvate from another source, well, here we want to get NADH into the cytoplasm to do gluconeogenesis. So remember, here we're kind of running the opposite situation. In this case, we can bring NADH or the equivalent of NADH, because remember that NADH itself cannot come out. So we're going to bring the equivalent of NADH. We're going to bring the electrons from NADH out of the mitochondria by hiding them in the form of malate. So we will take pyruvate to oxaloacetate with our pyruvate carboxylase in our mitochondria. Then we will take our malate dehydrogenase, our good old friend, and we're going to convert that oxaloacetate to malate. So we're using NADH. So this is going to be a reduction reaction. We're going to be transferring the electrons from the NADH onto oxaloacetate to give us malate. We can ship the malate out, and then when we're in the cytoplasm, we convert that malate back to oxaloacetate. So here we're going to be using malate dehydrogenase as well, but now we're using the cytosolic, and we're doing it in the other direction. We're going to be using NAD+. We're going to be doing an oxidation reaction. We're going to be regenerating oxaloacetate, and we're going to be regenerating NADH. And we're going to be regenerating the NADH by passing the electrons off of malate onto NAD+. And remember, those electrons that we took off from malate, they're coming from the electrons that we took off of NADH in your mitochondria. And so you've transferred electrons out of your mitochondria in order to do gluconeogenesis. But a lot of times you want the other situation. You want to actually bring the electrons from the NADH into your mitochondria in order to get them through the electron transport chain to generate energy. And so this is going to be what we'll talk about when we talk about the malate-aspartate shuttle. So to summarize, glycolysis is happening in your muscle cells and generating pyruvate. We're also generating 2-ATP that the muscles can be using directly, and we generate this to NADH. The NADH, when there's not enough oxygen with your muscles working so hard, the NADH can't go through the electron transport chain, but we can recycle it to NAD+ by using it to reduce pyruvate to form lactate. That lactate can then get shipped to your liver. Your liver will then use that lactate, take the lactate dehydrogenase in the liver, oxidize the lactate to pyruvate, regenerating our NADH. This pyruvate and the NADH can then be used in gluconeogenesis to make glucose, and that glucose can then get shipped back to your muscle cells. Similar things are happening if that lactate comes from red blood cells or any other cells. They can all enter the liver where they can get converted into glucose that can get sent back to your cells. So that was fermentation. Now let's talk, however, about when we do have oxygen, but the problem is our NADH is in the cytoplasm because we generated it from glycolysis or some other cytosolic oxidation reaction, and we need to bring it into the mitochondria, but the mitochondrial membranes won't let through that NADH. So we're going to sneak the electrons through because really the electrons are all that's needed for that electron transport chain. Doesn't matter where those electrons came from. Well, the only reason why it might matter is because how they get there matters in terms of how much energy you'll generate. But once they get there, where they came from, who cares? So let's talk about how we can get those electrons there. This is going to be our malate-aspartate shuttle and our glycerol-3-phosphate shuttle. Let's start by talking about the glycerol-3-phosphate shuttle. So this over here. The glycerol-3-phosphate shuttle is going to take electrons from the NADH, and it's going to bring them in by passing them directly to Q from this glycerol-3-phosphate dehydrogenase. So remember that when we talked about the electron transport chain, we talked about the kind of mobile carrier Q. And that Q, that ubiquinone and ubiquinol, basically what's going to happen is that's that carrier that takes electrons from either complex 1 or complex 2 and passes them off to complex 3 in our electron transport chain. We also talked about how there could be multiple other routes to Q. And one of the routes to Q is going to be this glycerol-3-phosphate dehydrogenase. Mitochondrial one is going to be FAD, use this FAD and FADH2. Well, your cytoplasmic one is going to use NADH and NAD+. And so remember, those were going to be like mobile soluble carriers, whereas your FAD, that's going to be membrane embedded. And so the mitochondrial version of this is going to be embedded in the membrane, and the cytoplasmic one is going to be free floating, and they're going to be working together. In your cytoplasm, what's going to happen is you're going to take that NADH that you generated in glycolysis, and you're actually going to use it to reduce dihydroxyacetone phosphate to form glycerol-3-phosphate. So this dihydroxyacetone phosphate, this is what we had in our glycolysis. So this was this step here. And so normally we're taking dihydroxyacetone phosphate that we generated from this aldolase step, and we're using triose phosphate isomerase to isomerize it to glyceraldehyde 3-phosphate. That's not a redox reaction, it's just an isomerization. However, we could also take this dihydroxyacetone phosphate and reduce it. And when we reduce it, then what we're able to do is we're able to generate glycerol-3-phosphate, which now holds on to those electrons. The glycerol-3-phosphate can now pass those electrons onto FAD in your mitochondrial membrane. And this is going to then we oxidize it, so we reverse this reaction. But here we're passing those electrons from glycerol-3-phosphate onto FAD to form FADH2, which can then pass those electrons onto Q. So here we're taking the electrons from the glycolysis, passing them onto NAD, generating NADH. We then pass the electrons from NADH onto dihydroxyacetone phosphate, giving us glycerol-3-phosphate. We now take the glycerol-3-phosphate and we pass electrons from it onto FAD to give us FADH2. We pass those electrons from FADH2 onto Q to form QH2, which can then take things through the electron transport chain. So we're using these components that were generated in glycolysis, so this dihydroxyacetone phosphate and this NADH, and we're going to use them to sneak those electrons into the mitochondria. What we note about this is we're taking those electrons and we're passing them directly to Q. When we pass electrons to Q, well, here, if we bypass complex 1, we're not going to be pumping the protons that we would pump out at complex 1. This means that we're basically only going to get the equivalent of 1.5 ATP per our NADH that we were generating in the cytoplasm, instead of the 2.5 that we would get if we were to go and insert that complex 1 with our NADH. So there's less of a yield here than we would get if we generated that NADH in our mitochondrial matrix, where it could enter at complex 1. And we'll talk about a method in a second where you can actually still enter at complex 1, even if you generate that NADH in your cytoplasm. This glycerol-3 phosphate shuttle is the main route that you're going to take to get NADH equivalents to the electron transport chain in your brain and in your muscles. But in other tissues, they're going to use this route called the malate-aspartate shuttle. And this is where we go back to that idea of that malate dehydrogenase we were talking about before when we were discussing gluconeogenesis. So this malate-aspartate shuttle, we'll see what here is going to happen is that we take oxaloacetate, we take the electrons from NADH, we pass them on to oxaloacetate, reducing oxaloacetate to form malate. We then ship the malate into the mitochondria, where we have a mitochondrial malate dehydrogenase that can take the malate, oxidize it to make oxaloacetate, and the process use NAD plus in the mitochondrial matrix to generate NADH, and that NADH is now in your matrix where it can enter at complex 1, and therefore you're going to get the 2.5 ATP equivalents from NADH that enters this way. So it doesn't look so bad if you just look at it the straight line. We're taking those electrons, passing them on to oxaloacetate to get malate, and taking the malate in. We can do this because we can't take the NADH in, but we can take the malate in. And so the malate is carrying those electrons into the mitochondria. I'm going to complicate things now to show you a more realistic view of what's going on, because unfortunately, there's a lot of kind of swappage that needs to happen in order for that malate to come in and in order for keeping the cycle going. In order to understand what's going on, we're going to need to review a couple of our concepts about membrane transport. Remember, we had that term where we transported two things through a single carrier. We call that co-transport. So we can have antiport, where things are traveling in opposite directions, or symport, where they're traveling in the same direction. Both of those would be forms of co-transport, and we could also have uniport, which is just going in this one thing at a time through. What we're going to see in our malate-aspartate shuttle is we're going to see antiporters. So we're going to take one thing in exchange for taking something else out. This is going to allow us to keep a balance in our mitochondria and in our cytoplasm that's going to allow us to keep the cycle going. So the key thing to keep your eye on is going to be this electrons that are coming from this NADH. Those electrons, remember, are going on to oxaloacetate to give you malate. The malate is going to get shipped in, and then the malate is going to be converted back to oxaloacetate with the conversion of your NAD plus back to NADH. This is all you really need to do in order to get the electrons in, but in order to keep things going, you're going to need to regenerate oxaloacetate out in the cytoplasm. You might think, why don't we just ship that oxaloacetate back out? But there's no transporter for oxaloacetate through the mitochondrial membranes. And so instead of shipping oxaloacetate out, we're going to have to disguise it and then transfer the disguised version out. We're going to disguise it in the form of aspartate, so that good old amino acid aspartate. How we're going to do this is using an amino transferase, in this case, aspartate aminotransferase, or AST. If you've heard about your liver enzymes, and like AST is one of them, and basically aspartate aminotransferase is this really important enzyme, and it is used as one of the main transaminases, where basically you take an amino group from something and you pass it on to something else to give you something else. So we'll talk a lot more about this when we talk about amino acid metabolism, but basically you can take any amino acid, remove its amino group, and pass it on to an alpha keto acid. So keto, it's got a ketone group, and it's in the alpha position to a carboxylic acid. That will generate a different amino acid and a different alpha keto acid. We'll talk a lot more about this when we get into amino acid metabolism. But here what's happening is we're taking an amino group from glutamate, and we're passing it on to oxaloacetate. This generates aspartate and leaves you with the remainder of alpha ketoglutarate. aspartate can get passed through. Aspartate can get passed through in exchange for passing in a new glutamate. So you take out the aspartate and you take in the glutamate that you need to do this reaction again. We took in the glutamate in exchange for the aspartate, where you take in the malate in exchange for taking out the alpha-ketoglutarate. So we need to remove the alpha-ketoglutarate in order to get rid of kind of our waste, and this will allow us to exchange it for malate. Our waste in the mitochondrial matrix, however, is not waste in the cytoplasm, because what's happening in the cytoplasm is basically we're doing the opposite of everything that we do in the matrix in the cytoplasm. So that aspartate that we shipped out, we can now take the amino group from that aspartate and we can pass it to alpha-ketoglutarate. This alpha-ketoglutarate is the alpha-ketoglutarate that we passed out of the matrix in exchange for passing in the malate. So we shipped out the alpha-ketoglutarate in exchange for taking in the malate. The alpha-ketoglutarate that we shipped out now can accept the amino group from the aspartate we shipped out, remaking the glutamate. And this glutamate we can then ship back in. All of this was regenerating the oxaloacetate that we needed in order to then do this again to pass electrons onto oxaloacetate from our NADH to generate our malate that we can then ship into the matrix. So basically all this fancy dancy stuff is to regenerate the malate. So let's review one more time what's happening. Let's look at it in a different view in case this one makes a little more sense to you. What we have out here is we have our cytosolic mitochondrial dehydrogenase. It's going to take the electrons from NADH, and this is going to reduce oxaloacetate to form malate. So remember, reduction is gain of electrons, our good old oil rig. So we're reducing oxaloacetate by passing electrons to it. This generates malate. Malate can enter the mitochondrial matrix in exchange for the passing out of alpha-ketoglutarate. This is happening through the malate alpha-ketoglutarate antiporter. So it's an antiporter because it's transporting these things in different directions. You're taking the malate in, and now a mitochondrial version of malate dehydrogenase can then take the malate and it can oxidize it to oxaloacetate. So here's where the reversibility of reactions comes into play, because we're able to take this, the same like enzyme or different versions of the same enzyme and do the reaction in different directions in different compartments of the cell. Here we're going to be oxidizing malate to form oxaloacetate, and more importantly, to generate this NADH in the matrix. So we've taken the equivalent of an NADH out of the cytoplasm and regenerated it in the matrix. Now this NADH, because it's in the matrix, it's able to go directly into complex 1, and so we're going to get the full energy out of it. If we want to do this again, however, we're going to need to take this oxaloacetate and get it out. But we can't take the oxaloacetate out because there's no oxaloacetate transporter. Instead, what we're going to do is we're going to disguise the oxaloacetate. What we're going to do is we're basically going to take an amino group from the amino acid glutamate. We're going to take that amino group, add it to oxaloacetate. This is going to give us the amino acid aspartate. And this is happening through this aspartate aminotransferase enzyme. That aspartate can now get shipped out. In order to get shipped out, it's going to go through this aspartate glutamate antiporter. As the name suggests, in antiporter, basically you're transporting two things in opposite directions, and it's telling us that we're transporting aspartate and glutamate. We're going to be transporting aspartate out and glutamate in. Glutamate, remember, is what we needed in order to get an amino group to pass on to oxaloacetate. This is going to allow us to generate aspartate. In the process of doing this, we're kind of left with the waste products from when we took an amino group off of glutamate. This waste product is our alpha-ketoglutarate. We can ship out the alpha-ketoglutarate in exchange for bringing in a new malate. So remember when we took the malate in, that was happening through the malate alpha-ketoglutarate antiporter. We shipped the alpha-ketoglutarate out, brought a malate in. And this is going to then allow us to get rid of our waste, bring in the new things we need. In order to make this a cycle so we can keep things going, we basically do the reverse of everything that we did in the cytoplasm in the matrix, everything we did in the matrix in the cytoplasm. So we can take this aspartate that we shipped out, as well as the alpha-ketoglutarate that we shipped out. We now take the amino group from that aspartate, pass it on to alpha-ketoglutarate. This is going to give us glutamate and oxaloacetate. The oxaloacetate is now ready to accept electrons from NADH. And that glutamate can now get shipped back in exchange for shipping a new aspartate out. So I know it seems really overwhelming when you look at a pathway like this, where there's a lot of complicated things going in. But the key takeaways are to think about what is the net effect. The net effect here is basically we're taking those electrons from NADH and we're bringing them into the mitochondrial matrix. Everything else is just to regenerate the system. The important thing is that we're the electron passing. Another important concept is that we can have reactions happening in opposite directions in different compartments or at different times. A final important thing is the idea of shuttles bringing in the equivalent of something when we can't bring in that thing directly itself. Also, the idea of antiport and introducing aminotransferases, which we'll talk a lot more about in the future. But this is going to come into play with amino acid metabolism, as well as when we think about the carboxylic acid cycle. Because remember, although we can break things up into cycles and pathways, it's the same sort of chemical logic happening, but that's going to be seen over and over again, which is why we did that glycolysis activity where we're really focusing on this chemical structure. So to review, glycolysis uses up NAD+, and it generates NADH, and that happens in the cytoplasm. If that NADH builds up, it's going to inhibit glycolysis as well as other catabolic pathways. To keep glycolysis going, we need to regenerate the NAD+, and there are a couple of main ways that we do this. If we have oxygen, we go through aerobic respiration. Because this is happening in the cytoplasm, remember, glycolysis is happening in the cytoplasm, we can use electron shuttles, the malate-aspartate shuttle, which involves malate dehydrogenase, and the glycerol-3-phosphate shuttle, which involves glycerol-3-phosphate dehydrogenase, in order to sneak the electrons from the NADH into the mitochondria. If we use the malate-aspartate shuttle, then you can get 2.5 ATP per NADH that you produce. And if you use the glycerol-3-phosphate shuttle, then you get 1.5 per NADH produced. Because here you're going directly to Q without pumping electrons, but with the malate-aspartate shuttle, you're able to go through complex 1 and therefore get the 2.5. That'll let you use the electrons to actually generate ATP. But if you can't generate ATP directly that way because you don't have oxygen or some other reason your electron transport chain isn't functioning, you still want energy. What you can do is you can take those electrons, pass them on to lactate to give you lactic acid with the help of lactate dehydrogenase. Remember lactic acid, lactate, those are just conjugate acid, conjugate base pairs. Both of them are a more reduced form than pyruvate. And when you reduce those, you have oxidized your NADH, you've regenerated the NAD+. The NAD+ can now be used to keep glycolysis going. Remember that NAD+ is needed by the glycerol-3 phosphate dehydrogenase, which is doing this oxidative phosphorylation of your glycerol-3 phosphate to give you this 1,3-bisphosphoglycerate. Often what we're going to be doing is this thing called the Cori cycle, where your muscle cells are going to be generating a bunch of that NADH because they're doing a bunch of glycolysis, and they want to keep glycolysis going even though they don't have enough time to get the oxygen to all their muscles as they're working really hard. So they're generating a lot of NADH, and then they ship it out to the liver, which is going to then, in the liver, convert it back to pyruvate. So when we talk about fermentation, what we're talking about here is kind of doing glycolysis in this anaerobic mechanism, this anaerobic manner, in which we go through this process, and then we reduce this pyruvate. So we use the NADH we generated to reduce this pyruvate. And when we reduce the pyruvate to lactate, that is going to produce this NAD+ again. And then we can use this NAD+ for glycolysis.